Plants require mineral elements for their growth and metabolism. Some elements contribute to plant structural components like proteins, lipids, and chlorophylls. Others have a role in osmotic, metabolic, and energetic functions of a plant. Except carbon and oxygen, which the plant acquires from air, and hydrogen from water, most of the other elements are taken up by plant roots from the soil. Hence, the type of soil plays an important role in growth of plants. Let us see how soil is formed and how the elements are made available to plants by the soil. Soil is formed by weathering of rocks. Based on the size of particles formed by weathering, soil types are classified as sandy, silty, and clayey. Formation of these particles either releases the elements into the water held by soil, or the elements are adsorbed on the surface of the soil particles. Organic matter in the soil is called humus. Humus also contributes to the elemental composition of soil since it provides large adsorptive surface for elements. Soil and humus particles generally carry a negative charge on their surface, which attracts positively charged elemental ions and retain them in the soil matrix. Soil pH plays an important role in making elements available to plant roots. For example, iron is readily available to plant roots in acid soils, while phosphorus is available in alkaline soils. The elements are made available to plant roots by mass flow or diffusion through the soil water. Absorption of the elements or ions by plants may occur passively by simple diffusion, through ion exchange at root surface, or by exudation of chelators by the root, which solubilizes the elements. Once absorbed by roots, the elements reach the xylem either through the water in intercellular spaces, or the elements may pass through the membranes of root cells and move intracellularly up to the xylem. Uptake of elements by plant cells is effected through channels or transporters present in plasma membranes. Channels show specificity for the ions that they transport, and this transport is passive. The transporters also carry ions passively, but are energized by a proton gradient created across the root plasma membranes by the activity of an H+ ATPase or PPase. Active transport of elements across membranes occurs by the activity of pumps, which can transport elements against a concentration gradient. In this module, we shall study the availability, transport, and role of three important elements, namely potassium, calcium, and magnesium. In soil, potassium is present as soluble ions in soil solution, or the ion is weakly adsorbed on clay and soil organic matter. Potassium content of soils is about 5 millimolar. Plants accumulate potassium so that its cytoplasmic concentration is 100 millimolar. Potassium uptake, therefore, takes place against a concentration gradient and requires energy. Potassium is transported along an electrochemical gradient present across membranes. Plasma membranes of roots have shaker-type potassium channels like AKT1, which stands for Arabidopsis potassium transporter 1, which allows inward flow of potassium ions, while AKT2 or 3 allow bidirectional movement of potassium across membranes of phloem parenchyma cells. Channels are also present in the membranes of guard cells or in vacuolar membranes. Besides channels, there are transporter families that transfer potassium using the energy from a proton gradient generated by H+ ATPases. These include potassium uptake permeases belonging to the HAK family of transporters, the HKT family of potassium transporters, and others. Rubidium ion is a potassium analog and shows similar size and permeability as potassium ions, but is not used as a nutrient by plants. It is used as a tracer to study potassium transport kinetics. It is seen that channels are used when soil availability of potassium is high, while transporters are used when potassium availability is low. Potassium plays an important role in many physiological processes, like maintenance of osmotic equilibrium in cells or causing rapid turgor changes that are important in bringing about stomatal opening and closure. The Cat1 or 2 channels are present in guard cell membranes and play an important role in potassium influx into guard cells that lead to the opening of stomata. GORC is an outwardly rectifying potassium channel which is also present in guard cell membranes. This channel brings about potassium efflux and causes stomatal closure. Persistent deficiency in soil potassium availability leads to development of characteristic symptoms like chlorosis and scorching of leaves along the margins. Older leaves show the deficiency symptoms first, since the potassium ions are mobilized from these leaves to young growing tissues. Continued potassium deficiency leads to growth retardation and death of the meristems. Plants show acclimation to potassium deficiency by various mechanisms like activation of high affinity potassium transporters, reduction in potassium transport through phloem, or through mobilization of potassium from vacuoles. Now we move on to the second element, calcium. Calcium content in soil arises from weathering of limestone. It forms insoluble salts like calcium phosphates, and hence much of the calcium is not available to plants. Calcium ions adsorbed on soil particles are taken up by plants through cation exchange. Soil calcium plays an important role in maintaining soil pH and influences the availability of other elements like iron and phosphorus. Plants adapted to calcareous soils are called calcicoles, and they show special mechanisms to solubilize iron and phosphate. Calcifuges, on the other hand, are plants that show sensitivity to the high calcium levels found in calcareous soils due to their inability to solubilize iron and phosphate. Calcium mainly enters roots through the apoplast. Influx of calcium into cells occurs through calcium channels. Cytosolic calcium is maintained at submicromolar concentrations. Opening of calcium channels leads to a rapid increase in calcium levels, which plays an important role in signaling. Cytoplasmic calcium levels are restored by the activity of calcium transporters that remove cytosolic calcium into the vacuoles or the apoplasm. Influx of calcium into organelles takes place by active transport involving calcium ATPases. An important function of calcium is that of a signaling molecule. Receptor proteins include calmodulins, calcineurins, and calcium-dependent protein kinases. Receptors transduce the calcium signal to subcellular compartments and leads to activation of proteins involved in ion transport, cell structure, and enzymes. It also brings about alteration in gene expression by modulating the activity of transcription factors. Calcium plays an important role in signaling, in regulating responses to development cues, abiotic stresses, and biotic interactions. Calcium deficiency symptoms include tip burn in sorghum or rot in tomato fruit. They are observed in young tissues because calcium cannot be mobilized from older tissues and redistributed via the phloem. Finally, we come to the third element. Magnesium is a constituent of basic igneous rocks. Soluble magnesium ions are present on the surface of clay particles and are available to plants through cation exchange. Magnesium ions enter plant roots through the apoplast. Magnesium transporters are present in plasma membranes of root tip cells. Transport of magnesium to xylem is by non-specific cation channels. A magnesium proton exchanger ATMHX is involved in regulating long-distance magnesium transport and is energized by an H+ ATPase. Magnesium is an important constituent of chlorophyll, and its transport to chloroplast occurs through a high-affinity magnesium transporter as well as a fast-activating chloroplast channel called FACC, which is located in the chloroplast envelope. Import of magnesium into vacuoles involves a magnesium proton antiporter. Vacuoles serve as stores for magnesium, and release of magnesium from the vacuolar stores has been shown to involve another transporter. Plant cells show magnesium concentration of 15 to 25 millimolar. Most of magnesium is bound to chlorophyll molecules. It also plays a role in joining and stabilizing ribosomal structure by interacting with the rRNA backbone. Magnesium serves as a cofactor for the activity of a large number of enzymes. Intervenal chlorosis is an important symptom arising due to magnesium deficiency. The symptom is first observed in older leaves, since magnesium is a mobile ion and is readily transported to younger growing leaves. Due to its role in regulating carbohydrate transport to sinks, magnesium deficiency leads to poor root growth. Given the importance of soil parameters and availability of important elements like potassium, calcium, and magnesium in soils for plant nutrition, it is important to carry out soil testing. Soil testing kits include tests for measuring water holding capacity of soil, its pH and conductivity, levels of important minerals in soil, and their available forms. Suitable manipulation of soil is important to ensure healthy crops.